image is your RNA, the transcription factors. Okay, so what is labeled in green color is the BRU, not BRDU. If you use BRDU and pulse label, that will label the newly synthesized DNA. BRU will label the nascent RNA. Okay? And it is interesting if you look at this particular picture, even other ones, you see that some crescent-like structures, right? That's because you have RNA, nascent RNA being transcribed around transcription factors. Okay, the central hollow part is actually the transcription factor. Transcription happens on the surface of transcription factor. Okay, so your transcription factor here would be in blue color. Red color is your nucleus staining. That's your transcription factor. Okay, so the second picture is your DU staining. Replication factors. You can also label DNA polymerase. Here to label interior, this blue part you can have RNA pol 2 phosphorylated antibody against RNA pol 2 phosphorylated, and it should be self-understood over here, right? In white color, like here is your nuclear, sorry, the causal body, one example of causal body. Nucleolus are over here, right? And the green color is your nucleus speckles. Now this is PML in green color over here. Is one PML body, and red color shows the protein inside. In this case, it's the BRCA1 BRCA1 protein is located inside the PML body. And remember, on the other day I draw the PML body like that, right? So then I showed that other proteins get post-translational modified inside, and you can actually appreciate that in this particular example. The rightmost image is DNA repair foci. So they have labeled H2AX phosphorylated at the sites of active DNA repair. You can see that all of them are making the nuclear condensates, nuclear compartments, subnuclear compartments, likely through phase separation. Okay, so we ended up our class, the last class on the slide saying proximity ligation basis, right? And as I said, proximity ligation means you are ligating the proximal DNA fragment, right? So how this technique is used to identify the especially proximal DNA segments inside the nucleus, or especially proximal protein molecules also, is actually based on cross-linking. Okay, so let us start with one of the technique, and this was given by Job Decker. In the early 2000s. Okay. The name of the technique is 3Cs, the abbreviation. 3C, the abbreviation. Chromosome information capture. Now, the original objective of Job Decker to develop this technique 3C was to identify the interactions between sister chromatids. What are the sequences responsible for sister chromatin pairing? Okay. That was the project, PhD or postdoc project that he was working upon. And in that process, he ended up innovating a new technique because there was no available technique in those days to identify such interactions. While developing techniques, he realized that it can have other applications. So the actual work which was supposed to be on sister chromatic pairing was not done through this technique. Since the other applications of this technique are much more appealing. Okay, so what we do in this technique is we take our cells, right? So you have cells either on a dish or in a liquid media.
इसलिए 2x भी अच्छा लगता है, तो अभी सुनती भी नहीं है।
And the first thing that you need to do is you need to fix it.
घर पे बैठ के दे रहा
As
होगा?
I mentioned briefly in the last class also, we fix it using formaldehyde. So 1% paraformaldehyde.
नहीं, मुझे नहीं पता, मैं कल की
And
नहीं
there is a
देखी,
reason
परसों की
that
भी नहीं देखी है।
why we use formaldehyde,
अभी
paraformaldehyde.
परसों वो
It
PCA
makes
बनाया
the
था।
crosslinks. Okay, it's a polymer. Okay, and it has some affinity towards certain amino acids and certain DNA residues, DNA nucleotides. So it is able to cross-link the protein DNA complexes, protein DNA, RNA, or the nuclear complexes, and fix it. Doesn't allow it to move, right? So basically, the moment you add paraformaldehyde, you're fixing the cells. The cells are dead, and you're taking a snapshot of each cell. Okay, so whatever cell cycle stage these cells were, it is fixed. Okay, each of these cells will be fixed once you add paraformaldehyde for 10 to 15 minutes. Okay, so it's a toxic thing. You might know if you drink. There was an incident where somebody had paraformaldehyde in a bottle and they went for some field work, and I think they kept formaldehyde to have the samples, live samples of fishes or lizard or reptiles and all that. However, it turns out that somebody mistook it and he or she drank it. Instant death. Okay, even with the 1% formaldehyde, if you drink a glass of it, you're gonna dead because it's gonna fix your molecules inside, right, inside your cells. Okay, so once you fix it, things are not moving now because that's the important thing, right? You can't start doing something and things were moving, molecules were moving. Then you do not know what was the exact conformation. Okay, it would have moved during your experiment, during cell lysis, and when you add other reagents, things are moving, right? So you have to first fix the things. Okay, so in this step, we are taking a snapshot of each cell and it is fixed. Cells are dead and whatever stage they were, it is frozen. Okay, it is cross-linked by PFA. The second step is, of course, you need to lysate the cell. Okay, so cell wall using detergent or whatever buffer you have, it is like you lysate it, right? So after cell lysis, you will be left with your nuclei, right? Now, cell lysis can also be performed using homogenizer, which is a physical method of cell lysis. And then you perform you can also isolate chromatin, or even before isolating chromatin, you can do the digestion of the chromatin inside the nucleus. Let us say you have actually isolated, just to simplify the things, digestion can also happen inside the nucleus. Okay, you permeabilize the nuclear membrane using some reagents and then add your restriction enzyme and it will do the digestion inside. Okay, so the next step is to do the digestion. Let us say you have isolated the chromatin after homogenizing to an extent that you, or using the chromatin extraction buffer, which will be enriched with the magnesium salt and all that. You have isolated your chromatin, which will not be completely intact, but good enough to work upon. Okay, and the first thing that we do on this is your chromatin, DNA protein complexes, protein complexes, and all. You do the restriction digestion. You add your restriction enzyme out there, and this will lead to complexes like that, right? Where you have protein complexes sitting. Protein complexes sitting in between, right? Since this is cross-linked, you will capture these complexes, right? Protein DNA complexed, and then after restriction digestion, you will get these fragments out from it. Okay, so restriction digestion is done. Some of you might say that why not other methods of fragmenting the chromatin? For example, sonication is a popular method for chip experiment. It turns out that each method has its own advantage and disadvantage. For example, sonication, the physical method uh, is more harsh. Okay, it will the transient complexes will be falling off by that method. Restriction enzyme is a much more gentle method of chromatin fragmentation. On the other hand, since it is a gentle method, it is actually capturing a lot of transient interactions which are actually noise. Okay, so sonication based method will have lesser noise. There are some versions of this method where people actually do the sonication. Okay, so each one has its own advantage and disadvantage. Okay, so what next? The next step is very important. In the original protocol, this was done in this way, but these days we do it in a different way. I will mention about it. So the next step is to perform ligation of these open ends. You see these open ends over here, right? These are open ends after digestion. So we need to ligate them. However, this ligation reaction is done in dilution. Okay. Can anybody suggest why in dilution?
Because we don't want to ligate the complexes and again form the original structure.
Well, close enough, but not entirely. So let's say this is the ligated products. Okay, and these are your junctions, ligated junctions. Sometimes both sides are ligated, while in other cases they are not ligated, partly because of the length constraint. Lengths were too small or too long, or they didn't. That was not optimal length to get ligated, or they were too short like this one. Okay, so you get this ligated product. Now this ligation, so ligation in dilution. The reason we do it in dilution because we do not want this kind of ligation, right? Cross product ligation, inter product ligation. We want intra product ligation, which are these ligation that we want to perform, right? That's why we do it in diluted form. If it is condensed concentrated form, then you will have cross product formation, which will be false products, right? So we got this ligated product done in dilution. The next step is you heat at around 60 degrees or so that your formaldehyde, which is keeping this complex together, will evaporate. So paraformaldehyde is a liquid state. If you heat it, it will convert into formaldehyde gas and that will evaporate. The moment it evaporates, you are de-cross-linking your, so this was your cross-linking. Cross-link. Right now we are doing de-cross-linking. Okay, and then the kind of products we got is these products, right? With these junctions. Okay, and this can be purified since after decross-linking, your solution in your tube will still have protein and other products which are not required. So you actually purify this, right? Purify DNA. So the third method is again your phenol chlorochrome method, right? Once you have purified that, you will have this in purest form, this DNA. Okay, so we are basically done. Now we need to understand what are we trying to do. We wanted to identify the regions which were proximal. Okay, regions which were spatially proximal inside the nucleus. For example, these two regions were proximal, right? To a protein complex, which could be transcription factors and other things. These two regions are proximal. Likewise, you can have many regions which were spatially proximal inside this nucleus. Let us say, if I zoom into this nucleus, and somewhere I had this scenario. There was a protein product in between. Okay, so and I knew both the regions. These two regions which are interacting in space inside the nucleus, I know them. Let us say I know this region, okay, and I know the other partner also. I know this region also. It's known to me, A and B, okay. Now you can think of what is it. A could be your promoter, B could be your enhancer, right? If enhancer promoter are interacting, if promoter to promoter are interacting at the transcription factory, then A and B are two distant promoters. I think it will be helpful if you people actually speak up rather than typing. Anyway, so if this is the scenario, these products would be present somewhere in your tube, right? All this is happening inside your tube, right? All these assays are actually happening inside your Eppendorf tube, right? So somewhere here it's like this, right? So you will have these products inside in the following form. One of them is actually like that, right? Circular one, this one. Other one is like that. Another one is like that, right? And these are junctions, right? Somewhere you will have this is coming up. This may appear somewhere. Let us say it is appearing over here. This was your A partner over here. And this was your B partner. If they were interacting, there might be a case that your hypothesis was incorrect, that they were not interacting. Let us assume that there was a hypothesis and it turns out to be a true hypothesis. They are together. Now, they will come like that next to each other after ligation, isn't it? So let us trace back these fragments. Where will they be? Is here. And then they were here. Right? And let us see. And let's say that those are one was here and another one was here. Okay, they were spatially proximal, and we can trace back, and they are together inside the nucleus in the following form. Right? Okay, so your tube, your Eppendorf tube will have this cross product form, right? This ligated product form where you know both the partners. Now, what can you do to identify this, that whether this product is formed or not? What will you do? Simple thing, simple essay. No, just you know A and B. So why will you go for sequencing? Sequencing will cost you a lot. You know A and B then, right?
PCR?
Yeah, just if you know A and B, you can always get the primers from A and B regions, right? Convergent direction, and you can amplify this. So what will happen is the following: you will have amplified product.
We can get the junction PCR done.
What is junction PCR? Yeah, junction PCR, but then why there is no need? Why to make any junction primer at all? You know A and B, and you amplify. If they give you products like this in PCR, it means they were together in the nuclear space like this one, right? If this product is not formed after PCR, you're not able to measure in your CT value, you're not able to measure this thing, okay? Then it means A and B were not together, isn't it? Now you might say that, well, I got some quantitation through quantitative PCR, but I don't know whether it's significant or not, the presence of AB product. I did see something, it's not like I don't see, it's not like my CT value is really very high that there's no product out there. I see some kind of intermediate thing and then I'm confused whether it's present or not. If it is present, this interaction, is it present in most of the cells or very few of this? What is the interaction frequency in terms of number of cells, right? So then you have to look at the control region. So what you could do is the following thing. What could be your ideal control? Your ideal control could be the known interacting partner in literature. Okay, that's your positive control, and known negative interacting partner, which is known not to interact. Okay, that's one way of doing it. In the beginning days, people were doing this. But one of the easiest way or straightforward or convincing way to do the same thing is the following. Look at your locus. Let us say I was checking enhancer to promoter interaction or promoter to promoter interaction. This is your enhancer and here is your promoter. Okay. And this is your, you can say that this was my red color. And that's my green color. Okay, I wanted to check whether the interactions of these two were significant enough or not. So through PCR, I look at the number of copies on the y-axis, right? So quantitation, what do you say, quantity, you can say. Inverse of CT value, whatever you do, or number of copies, you can say. And as a control, let us say you fix P, promoter is your fixed, and then you want to see whether it's interacting specifically, this promoter is interacting specifically with this locus, okay, and not the neighboring locus. Then what you could do, you can take a lot of the neighboring loci upstream to the candidate of interest and downstream, and look at interaction of this promoter with all these loci. What you could find is the following. If I now plot the interaction frequency of this promoter to this enhancer or this promoter to these other loci on this plot, then I might say see that there was some baseline thing and then it is shooting up right at enhancer and again it dropped up. Right, and of course, since P is my bait region, which is common for all of these interaction, I'm looking at this promoter to all other loci. Okay, so then I know that these two are interacting, right? And rest of the neighboring regions are not interacting, so they are my negative control. That's how I know that this enhancer promoter proximal in a 3C assay. Okay, is that clear? Okay, and somewhere I said that people are also doing this assay, restriction digestion as well as ligation inside the nucleus also. That's possible. Okay, that's called in situ 3C. In that case, you don't need ligation in dilution because now you are doing So it would look like following. Let's say this was your nucleus, okay, and inside the nucleus you have digested the chromatin through pores, right? You made pores out there, let's say pores through certain buffers, permeabilizing agents, and so on. You don't disrupt it completely, unlike other assay where you isolate the chromatin. Okay, and restriction enzyme has digested the chromatin, so it would look like this, right? Inside the nucleus, lot of fragments, but native position is likely to be preserved, right? And then you add your ligase enzyme inside, so these regions will be ligated, right? So in situ, this is called in situ 3C. Okay, so but this was not the original assay. Original assay was having this smart technique of ligation and dilution. The problem is the observation and the practical observation. Many people in the field, what was the need of in situ 3C? Because people observed that when they were taking the diluted restriction digested product for ligation, okay, they were diluting it. They took some sample through pipette and look under the microscope. They can still see clumps, okay, which tells you that there is still a very high possibility of cross product formation. Although the conceptually, theoretically, it shouldn't form if your dilution was properly done, but still some products are stuck that even dilution is not able to detach them. Okay, so that's why in situ 3C came up. Concept is same, it's just that ligation and dilution is not did everything you do inside the permeabilized nucleus. Okay, now some of you might be wondering what we started with was something else. I said that we're going to identify all the interactions in the genome, right? What all genes are coming together. That's what the question we asked in the transcription factory concept. Now, right now, what we did was very different. We just tried to look at whether A is interacting with B. Two known regions are interacting with B. So that's what the scope of this technique is, that 3C tells you one-to-one interaction, okay? One-to-one. That's the scope of 3C. Very limited scope, but nevertheless, it was quite useful. And originally what Jock Decker did, he took one of the chromosome of yeast, which is smaller one. Okay, so what he did was he took one of the yeast chromosome and did number of 3C experiments of by taking different regions here. So he took some bins, uniformly separated bins of each chromosome, and he did the 3C in various combinations, all-to-all combination over here. And so on, right? So if you do so, you will get to know the structure of this chromosome. It turns out the structure was crescent-like structure like this, where telomeres were co-positioned together and centromere was away. Okay, crescent-like chromosome structure. Now you might wonder, how did we go from 3C to chromosome structure? We will talk about this little later. Otherwise, some of you will complain that this is again bouncing over. Okay, chromosome structure. However, you can do that, and you will see that there's no reason to get surprised. Over the time, I will elaborate the method in little detail. Okay, but right now, let us move on. So this was the original paper published in Science in the year 2000 by Jock Decker. Okay, so he used this technique to solve the structure. For the first time, chromosome structures were solved. Okay, so let's move on now. This was 3C. The problem with the 3C is the scope, right? So if I write technique here, and then I write scope here, 3C, the scope is just 1-to-1, right? We wanted to move on. Many people had this question that I know my locus. Okay, let us say I know my promoter. One of the question could be I know my promoter. I want to see what all enhancer it is having. Okay. Now you might say that is this the way enhancer function through three-dimensional looping? Well, yes, that's the present view. There are a few conflicting observations that we will discuss later on. But let us say I know my region and I want to see all the interactions in the rest of the genome. Or I suspect that I know one enhancer or some locus control region or super enhancer, and I think that it has pleiotropic role. It has many promoters it is regulating. The same enhancer, many different promoters, same promoter, many different enhancer in the rest of the genome, which means that I need to find out one to all. Okay, this is my known thing. Okay, in this case, both of these were known. Okay, so now I'm trying to look at one, two, all interaction. One is known to me. I want to identify rest of them. So the answer to this was 4C. Okay, now this is the extension of 3C, logical extension of 3C, and there were two papers which came together. One of them called it as circular 3C. So that makes, if you add up this C now, it becomes 4C, right? 4C. Another fellow called it as 3C on chip, chip-like microarray chip and all that. Okay, so this C he added to 3C and it became 4C. They have different, same abbreviation but different full forms. Okay, so we still have time, so let's discuss the 4C now. How do we do it? A lot of the steps I will skip because you already know 3C. How does it work? Let us say that I start from the step when you when you did ligation. Okay, so your ligated products were like this. Now I say that I know one of the region now. Okay, so I can't color red and green. I can only color red. It turns out that if I have my red color coming over here, red color means known region. Okay, this region was known to me, and I want to identify this one. It turns out there is no way I can do this with this product that I have formed here. Okay. However, if I do the same thing with the circular product, if I do the same thing with the circular product, it is possible. This is known to me, right? And I want to identify the unknown one. That's possible. So what you do, you ignore these kind of products. Okay. And you might say that, well, if my known region only is this kind of thing, then it's not possible to capture the circular one, right? So you make sure in the beginning that your locus is, have a reasonable length. How do you know? Let us say this was your locus, and here is your region of interest. Okay, region of interest. So you choose a restriction enzyme which does cut sufficient length. Let's say it is cutting from here, restriction enzyme. Okay, it is a it has sufficient length to actually have this form. Although we cannot avoid this thing, this will also be there. But make sure that you had sufficient length of your bait or the reference region that your known region, so that it has tendency to form the circular product. Okay, and then if you had large number of cells, okay, and many cells have this circular product, that will be, and you also have appropriate control, it will be smoothened out, right? Uh, if there is a fluctuation that, let us say, 1 out of 20 cell is having this circular product form, others are, due to some dynamics of DNA fragments, is not making circular product. But if you have sufficient number of cells in millions or billions of cells, then that number will shoot up, right? So you will have sufficient DNA in hand to work upon. Okay, so the idea is to focus on the circular product. Idea is to focus on circular product. Okay, so this was after ligation. So what we do now is, we have done the splitting thing, decross-linking thing, right? Dx and purification, DNA purification, and then I have two kind of products now. One is your circular one, and others are your this kind of products, right? Ligated one that will be there in your sample, right? And this one is having one region which is known to you. Okay, and this will in your tube, this will not be just one region, right? So you are doing this in a population of cell, right? So you had a cell population from which you isolated all this DNA. So this is just one thing that I'm showing. Okay, although there will be several such circular products, right? Some of them will be redundant, others will be non-redundant in terms of the ligated product. Each one is coming from different cell, right? So this is not known to us. What is this unknown one? This could be different one, right? I know my region, this one. So how can I identify these unknown partners now, which are out there in these circular products? In all these four circular products that I have drawn, I know the red one. The question mark tells you that those partners are different partners, but I don't know them. It could be 4 different partners, or 3 different partners, or 2 different partners, or just one coming 4 times. Let me color them in different colors, right? So one of them is green over here. Another one is blue. And yet another is yellow. Okay. Anyone, what will I do to identify this green, blue, and yellow ones, and the white one over here? Well, I can again do PCR, right? I can do PCR, but now it's not convergent PCR, it's the divergent PCR. I will do other way around. I know the known region, so I can get the primer from this locus and from this locus, right? Get the primers and amplify these unknown ones. So now this will be amplified, right? You can amplify this one. And likewise, you will amplify your green ones, blue ones, and the yellow ones, right? You can amplify that. Of course, the terminals of these, the terminals will of course have that primer sequence from where we got the primers, right? From the known region. This is the known region. Red color is the known region because the primer is also included in the amplifier, right? Primer position, primer sequence. So I have now amplified the unknown products, isn't it? So from where did this idea come for the divergent PCR, not the convergent traditional PCR? It's coming from the plasmid thing. Sometimes people want to amplify the plasmids and they have their known gene inserted in the plasmid. So they know their known gene, but they don't know the sequence of the plasmid. So they take the a divergent PCR to amplify rest of the plasmids to get to know what the plasmid sequence was. Right. So from there, this idea translated to 3C and it became 4C now. So now, since we have all of these, so all of these will come in one tube, right? All of this is in one Eppendorf tube that, or PCR tube, right? PCR tube. Right. So all of them are present out there. What you do is just do the sequencing of this next generation sequencing. You can also do microarray, but for example, you have a chip and each spot tells you one genomic region of the genome. Okay. Then you take your sample, this amplified sample, and pour it over this, and corresponding complementary DNA will hybridize over there, right? So, for example, green one is hybridizing over here. Then you take the image and look at the relative signal at each spot. So you can actually uh, intensity will tell how much product was there at each spot, right? How much unknown region was ligated to your known region by looking at the intensity of these signals, right? And that's what the 3C on chip is. 3C on chip thing. Originally it was like that, but now you don't actually need to do this. Just do the next generation sequencing. Okay, so what you do, you add the universal adapters and send it for sequencing, or if you have sequencer, you can do it yourself. I took few classes on NGS for genome structure course, right? So, or you can refer to those lectures. So this is the way we identify all the unknown partners. So the scope, for example, I will also share some studies done on using 4C. So in a chromosome, you have one region of interest like this one. You can get to know where all it is interacting. Okay, will be high frequency interaction in the proximity, right? And then you will have distal interaction with less frequency, and you can again plot the signals. Signals would be, let's say, like that here, little bit more intense here, more intense, and this is the gate customer region like that. Right, so these are your signals out there, and there's some statistics goes into analyzing this that we can discuss in the next class. Not only this, you can actually identify the interactions on the other chromosomes, trans interactions, right? If this is interacting with the loci in some other chromosome here and some other chromosome over here and so on. Okay. So we will start from this slide in the next class. How do we really analyze this thing? I will not get into great detail, but I will tell you the basic idea of data analysis for 4C. Okay, so that was all for today, and we will see in the next class that this scope thing that I'm talking about, this scope thing can go to many, not all to all, many to many. Okay. So many to many is one thing. That is known as 5C, okay, which stands for carbon copy 3C. Okay, we will see why do we call carbon copy. And then we will have all to all in the end. In the end, we will have a method for all to all interactions. So it will be all to all, whole genome to whole genome. That's called high C, high throughput conformation capture assay. Okay, so I'll stop here. If you have any question, I can answer those.
Sir?
Yeah.
Sir, since here the 4C protocol is based on different cells, so will the cell-to-cell variability be an advantage or disadvantage here while in our aim of capturing one-to-many interactions?
Well, if it is coming from multiple cells and your number of cells that you started with are millions or billions, then you get the average picture, right? What you can say that on an average, this is the scenario in this cell population. If you want to do per cell, then you need to opt for single cell assays. Okay, and there are single cell high C and 3C assays available. Okay, so where you those are very sensitive assays where you only look at one cell at a time. Okay, and then you have typical instruments to do so. First, you isolate single cells and do the things in a very different way. But the underlying concept, which is the proximity ligation thing, is the same. If you get that kind of data from single cell high C, you can calculate cell to cell heterogeneity. And of course, it will be heterogeneous. I'm yet to see papers. Okay, there's a buzz thing these days of single cell genomics, single cell transcriptomics, single cell high C, and so on. Everybody says in this cancer cell population heterogeneity, this heterogeneity, that is pretty much expected. Yeah, heterogeneity without doing single cell experiment, I can tell you that there will be cell to cell heterogeneity. Why? Because each cell has its own individuality. Because uh, look at the cells which are, if there is a colony of cells, let's say this is a colony of cell, okay, or maybe a tissue. There are cells which are actually peripheral to this, which is exposed to the outside environment, right? And then there are cells which are inside. So environment itself is different, right? For each cell, which cells are in the neighborhood, what all proteins they are secreting, what is the physical stress they are experiencing. That also changes the transcription landscape and the chromatin interaction landscape. So when you say heterogeneity, well, I would say it is expected. I want to see a study which shows lack of heterogeneity uh, for the sake of uh, for the sake of change. So it's advantageous if you can show that this heterogeneity in chromatin interaction explains certain phenomena. If you don't explain anything, claiming heterogeneity is anyway expected. And I think very few people have shown that how this is actually explains certain kind of phenotype, or certain kind of phenotype people can explain certain kind of outcome of that interaction. Okay, whether the transcription was also showing stochasticity and heterogeneity or not at the same time in the same assay. So very few studies have shown that.
Sir, and since proximity ligation is the whole concept underlying all of these assays, is there any other concept by which we can determine the structure?
Yes, there are others. So I will talk about that later on. There are methods where you take the nucleus and take the sections of the nucleus and find out what is the content, DNA content in each section. So we'll talk about that method later on. These days, microscopy-based method, multiplexed DNA fish methods, they are also picking up, where you use many different probes spread around the whole chromosome to find out the structure. But these are good for visualization, right? If you want the precise coordinate information, IC-based methods are good. Of course, things are keep evolving all the time. People are always looking for better methods.
Sir, just a minor follow-up. So you said to find the coordinates and all. So how does one do that? Like, what is the reference to find the coordinates of loci?
Well, you know the genome sequence, right? You know these coordinates over here.
Oh,
So,
okay.
so let us say if I tell you that which gene is interacting with all genes, then you will look for physical position of those genes, right? So if you look at some of the not all, some of the microscopy based methods, they are not good at resolving this kind of resolution. For example, either you can just do 2, 3, or 5, 6 loci and look at their known loci and look at their interactions and calculate frequency by counting the cells in which they find the proximity. People take the whole chromosome painting, right? Then you can find out how the chromosome looks like in 3D, but you do not know which gene is where, right? So these methods were the first few methods which were actually telling you here is your gene located, and this is interacting with this gene, this coordinate is interacting with this coordinate. Now, as I said, there are methods coming up which are trying to do this multiplex thing, multiplex DNA fish, but those are very tricky methods. Very few labs are, only 1 or 2 labs are doing this because they develop those assays. They also have their own limitations in the resolution. In this case, the resolution will be determined by the choice of restriction enzyme. For example, if you use the restriction enzyme of fork cutter, which means the restriction enzyme is recognizing, identifying DNA motif, which is 4 base pair long, okay, GATC only. Then the resolution would be very high. But if you take the 6 cutter enzyme, where restriction enzyme is recognizing it got disconnected. Yeah, so what I was saying is that if you use 4 cutter enzyme, the average restriction fragment size will be 256 base pair. If you use 6 cutter enzyme, it will be 4096 base pair, theoretically, right? Average value will be around this. So as far as resolution is concerned, these assays gives you very good resolution. So that was all for the day.
Recording stopped.